There are a number of novel genetic technologies that are being developed to improve human health, both to be able to counteract the effects of genetic disease as well as to prevent the spread of zoonotic diseases into human populations. Even though these are really newsy and exciting ideas to think about, they have at their basis a kind of genetic interchangeability that has been true throughout the whole history of life. The first attempts to develop recombinant DNA technologies dates all the way back to the 1980s. One of the first such experiments was to introduce a gene that was taken from a firefly called luciferase and inserted into the genome of the tobacco plant. This use was to make the plant have a so-called reporter effect, given that certain conditions in the cells were true. In this case, they were programmed to glow when the tobacco plant was infected by a virus called the tobacco mosaic virus. One thing that you might not know is that there actually is a lot of tobacco grown out in western Massachusetts. The leaves that are grown there are used for the wrappings around cigars rather than the typical burn-in tobacco. A number of years ago, I actually had a student whose job out in western Mass was to help to maintain this crop. What she would do was, when the moon was new, go out into the field with other workers and pull up all the tobacco plants that were growing. This way, they were able to remove the infected plants from the field in order to protect the healthy ones. Recall from the last lecture that there are a few different varieties of genes. We remember that genes are segments of DNA that code for a particular protein. Most of the genes that we care about are our so-called protein coding genes, ones that actually carry the information for a functional protein. Enhancers are segments of DNA that tend to increase the expression of DNA. Expressing DNA is a euphemism for causing a particular gene to get transcribed and then translated into proteins. Another variety of genes, once they have been mutated sufficiently, are called pseudogenes. Pseudogenes aren't actually used for making their protein product anymore, but they're still copied by the cell when mitosis happens, and if they are in the germline, those cells that make the sperm and eggs will pass those pseudogenes down to offspring as well. Another thing to remember from last time is the different kinds of point mutations. The slow, stepwise mutations of genes is very common, but the effects of the phenotype are not the same. Silent mutations are changes to the DNA that do not result in a change to the protein, so there's no change to the phenotype. On the other hand, non-silent mutations are ones where there is a change to the protein. This might be a significant change by inserting a stop, therefore truncating the length of the protein, or it might be one that confers some kind of selective advantage for that particular trait. When we are thinking about natural selection, we have to bear in mind a couple of different aspects. First, we should consider the strength of selection on the trait. On the left side of the slide, you see that this population has a range of body size. We can differentiate the strength of selection in a couple of ways. Strong selection is the case when only the largest individuals reproduce, and weak selection is when only the smallest ones don't reproduce. In cases of strong selection, there tends to be a noticeable change in the average value of that trait in the next generation, but for weak selection, the difference in the average value of that trait tends to be a lot smaller. In addition to this, natural selection can be driving in a few different directions. The evolutionary trees on the right show 3 hypothetical examples of types of natural selection. Each of the dots on those trees represents a mutation. A gray dot is a silent mutation, and a colored dot represents a different non-silent mutation. For neutral drift in the middle, notice that there are an equal number of silent and non-silent mutations. Sometimes protein changes, sometimes it doesn't, but it really doesn't have an effect on the fitness, so you see equal numbers of proportions of each mutation in each lineage. In purifying selection, only silent mutations are tolerated. This would be the case for genes that are very tightly fit to their environment, and any changes to them are going to tend to decrease their fitness. Here's an example. Richard Lenski at Michigan State has a landmark long-term experiment on evolution taking place. The experimental setup is shown on the left. He started with a single strain of E. coli, which is a common bacterium in the human gut. He then distributed that sample to 12 flasks that were composed of genetically identical strains and let them grow. In the morning, at the beginning of the experiment, he gave each flask a supply of glucose, which is a simple sugar food that the bacterium lives on. And by the afternoon, the glucose would run out, and then the next morning, he or one of his students would take a small sample from one flask and give it a fresh supply in a new flask, and then the next day you repeat this, and every day forever for more than 30 years. One thing that's great about model organisms like E. coli is that they allow us to study long-term effects of evolution in simple living things that have fast generation times. In optimal conditions like here in the lab, E. coli can have a new generation every 20 minutes. This makes them a lot more effective to work on in evolution than if you were trying to study evolution in people or elephants or whales. Another smart aspect of this research program is that you would also freeze a sample of each of the 12 lines every 500 generations, making a sort of fossil record of his experiment. And this is even better than a real fossil record because those samples can be unfrozen later and revived as though nothing happened. So you have real living snapshots of the genetic and phenotypic condition in earlier generations. The graph on the right side of the slide is showing the relative fitness as generational time has gone on. Looking at our axes, notice that the x-axis starts at time 0 when the experiment started and goes out to about 20,000 generations. The other axis shows mean fitness, meaning about how this reflects the reproductive success of the bacteria in those samples. Over generational time, notice it started from the baseline of 1 for their measure of fitness, but nearly went up 50% over 20,000 generations. This is what purifying selection would look like, an organism in a really stable environment that shows progressive honing of its phenotype for the environment it finds itself in. This is also what Darwin would have predicted too, that natural selection would work slowly and gradually over many generations. But it is necessary for us to consider the heritability of the gene on how the phenotype presents itself. Again, looking at strong selection on a trait with a normal distribution like body size on the left, a trait with high variability and a strong selection would result in a subsequent generation with a large change in the value of the trait. On the other hand, traits with low heritability, as on the right, show that even with strong selection for one extreme of the phenotype, there is not a noticeable change in the average value of that trait in the next generation. Here are a couple of examples. The beaks of Galapagos finches have been adapted to crack particular kinds of foods. Peter and Mary Grant at Princeton University have undertaken a long-term study of the finches in their native environment in the Galapagos and have developed some notable records on the shape of the beak of these finches and how it changes with time as climate changes in a given year. On the left, this graph is comparing the average depth of a parent's beak with the average depth of their offspring's beak. This positive relationship indicates that ordinarily parents with deep beaks have offspring with deep beaks too. The bar chart, labeled 1976 adults, shows the proportion of beak depths for all adults sampled in that year, and the colored proportion of each of those bars shows the individuals that actually made it and had offspring later. Notice that the average size of the beak for that whole sample was around 8.8 millimeters in 1976, but it was mainly individuals who had larger beaks that had successful broods that year. What happened when it was mainly individuals with deeper beaks who became parents? In the next generation, the average beak depth of their children was closer to the 9.8 millimeters that was typical of their parents. So, this was a trait that under selection, with a high level of heritability, found that the average value of the trait in the population changed noticeably from the parents' generation to the next generation. But what does it look like when heritability is weak? My favorite example is this cute one. Seal point Siamese cats are born with dark noses, dark ears, dark tails, and dark feet. The adults continue this, although their coat will get a little bit darker, and in very old Siamese cats, they become more and more brown or black. The thing that's interesting about this is the pattern of their coat color is due more to the conditions of their environment. The fur gets darker on the parts of their body that are colder. Cold temperatures activate the melanin-producing function of melanocytes in the fur. Siamese cats that live in warmer places tend to have more white, but Siamese cats that live in colder places or ones that are older with slower metabolic rates so they're a little bit colder overall, tend to have more black or brown in their coats. So in this case, the heritability isn't that great, and there's a greater environmental contribution to the phenotype. Every trait has a so-called gene by environment interaction, and this is one in which the environment makes a stronger contribution to the phenotype. Okay, so far we've been talking about stepwise, point mutation-like changes to genes. Now we're going to talk about where the big money happens with phenotypic change: gene duplication and recruitment. The pattern works like this. First, let's take a given protein coding gene and whatever upstream regulatory regions will regulate its expression. The first step is to duplicate that gene and its regulators into two identical regions of DNA. At this point, there is no change to the phenotype because there's been no change to the information of the DNA. But the thing is, this is like when I used to go get ice cream with my children when they were little. They would ask if they could get the ice cream in a cone, and I'd say, sure, but let's get a cup too. This way, in case there's some kind of mishap, you don't lose the ice cream altogether. What this means for genes is if there's a change in the regulatory regions, then the gene doesn't get expressed and turned into protein at the same time as it did before. That's okay, however, because the original gene is still there, so you have this safety factor built in. The protein coding gene can also mutate, and there are a few different fates that can happen to it. First, one possible outcome is that the gene will mutate into a pseudogene that doesn't have any function. In this case, the DNA is still faithfully replicated during cell division, but it's just dragged along to no particular benefit or harm to the individual. Another possibility is that the second copy can mutate so it specializes in a reaction that's related to the original gene but not identical to it. In this case, you might have a gene that can be co-opted for a separate but related function from the original protein's phenotype. Yet another possibility is that duplicated gene evolves a new regulation pattern and is used for a different function altogether. So, unless mutation turns this gene into a pseudogene, we have a couple of different outcomes in which we have novel genetic information that is inherited in large chunks rather than in little stepwise point mutations. This probably results in the greatest amount of evolutionary change that we see among living organisms, and this is something that took scientists a long time to understand. Let's look at an example of how we see this working in real life in a laboratory experiment. Going back to Lenski's long-term evolution experiments, I added a plot to the slide from before. The x-axis is still generational time. On the y-axis, the optical density is actually a measure of the cloudiness of the solution. You can interpret this as meaning more bacteria in a given volume of the fluid, meaning that they reproduce more. So basically, it's a measure of fitness for our purposes. Now here's the thing to notice. We are zoomed in on a period of time after the plot shown up above. As you might have predicted from the trend of the upper plot, that mean fitness line was starting to flatten out, and when we see it at generation 30,000, it's looking pretty flat, but then at around generation 33,000, there's this big jump in fitness. What happened? As you might have predicted from the trend of the upper plot, that mean fitness line was starting to flatten out, and when we first see it at generation 30,000, it's looking pretty flat. But then around generation 33,000, there's this big jump in fitness. What happened? What happened was gene duplication and recruitment. As I said before, these bacteria live off the simple sugar glucose that's in the solution that they're swimming in. There's another substance that's also included in the solution that they live in called citrate. Citrate is a derivative of citric acid that can be used as food for these bacteria, but you'll notice in the upper picture that this particular citrate enzyme is only turned on when there's no oxygen. For the bacteria, this would mean that later in the day, when they've used up the oxygen in the solution in their flask, it would be unable to use the glucose for food, so they'd switch over to citrate and be able to break it down without the oxygen. What happened was a segment of DNA was copied, and that new promoter was able to cause the gene for citrate to get turned on even when oxygen was around, effectively doubling the opportunities that these bacteria had to bring in energy. How was Lenski and his crew able to figure out what happened? They unfroze some of their fossil record samples and let them do their thing for a while. They found that the same mutation happened in the same way when they ran it a second time. This really clever insight was made possible by the development of this really complicated experimental design with a lot of foresight. But is this something that only happens in the laboratory? No. Here are a couple of examples of how we see gene duplication and recruitment involved in making novel phenotypes in animals. First, venomous snakes. All venomous snakes have adaptations that allow their salivary glands to have been modified into making venom that gets injected through a hollow fang into their prey. Get ready to pause the video for a second and think about what other kinds of venomous animals you can come up with. If you're like most people, you probably came up with an animal on this list. Most of these are good examples, although that cute little poison dart frog is actually poisonous, not venomous. What's the difference? If it bites you and you get sick, it's venomous. If you bite it and you get sick, it's poisonous. Now, I know that's a fine point, but it's one that'll make you look smart, and that's what I'm all about. The Portuguese man-of-war actually isn't a good example because it's venomous, but not in a homologous way that the snakes do it. In contrast to the snake's venom apparatus, the jellyfish have these little stinger cells that are triggered when something brushes against it, and then the barbs and thread inject their venom from there. It's a cool system, but a different one from what we're talking about here. Actually, there is one kind of venomous mammal, and it's the duck-billed platypus. The males fight with each other for access to females, but instead of having a fang attached to a venom gland, they have what appears to be a co-opted scent gland that is attached to these little spurs on their hind limbs. Interestingly, they do share a lot of genes in common with snakes to make this happen, but snakes have really super duplicated it to a great effect. You can pause this for a sec if you want to dig in a little bit, but the take-home message is this. All of these different genes with names like ADAM or crotamine or acetylcholinesterase or BNP are all duplicates of earlier beta defensin genes that were used for defense against infection. Each of these proteins today causes different things to happen that will kill cells, and together they make up the cocktail made by the cells of the venom gland. So over evolutionary time, we have seen that early tetrapods had immune systems that used these beta defensin genes to be able to make proteins to prevent infection. Mammals have some of these, but in the case of the venomous snakes, those genes were duplicated and became specialized for potency and in slightly different functions, ones that made that cocktail of the venom made by the venom apparatus. In The Origin of Species, Darwin uses kind of a Socratic method to make his argument. What he would do is try to come up with an issue that the reader might have with his explanation, and then unpack why that explanation isn't better or isn't applicable to his argument. In this chapter about difficulties on theory, he tackles the question of what he called organisms of extreme perfection. He says, to suppose that the eye, with all of its inimitable contrivances for adjusting the focus to different distances, for emitting different amounts of light, and for the correction of spherical and chromatic aberration, could have formed by natural selection seems, I freely confess, absurd in the highest degree. Basically, I see how this is hard to imagine, and shouldn't that discredit my whole idea? This is a classic move that we see when creationists try to fight with Darwin, where they reproduce part of his argument, but not actually the part where he explains it. I think this is equivalent to giving the setup for a joke, but neglecting to tell the group the punchline. And here's the punchline. I'm not going to read this whole thing to you, but I highlighted a couple of things to get the main gist that he says, that the old saying of vox populi, vox dei, as every philosopher knows, cannot be trusted in science. Or just because everybody says it's true doesn't mean that it's true. He also says that the difficulty of believing that a perfect and complex eye could have been formed by natural selection, though insuperable by our imagination, should not be considered as subversive of my theory. This means that even though you might have trouble imagining it, it doesn't mean that it's impossible to happen that way. Now, 150 years later, we can look at something extremely complicated like the eye and be able to explain it developed in a stepwise gene duplication and recruitment kind of way. When you look at the structures of eyes in mammals, you'll see two main families of genes that are duplicated for different functions. The crystallines all form what are effectively the lens of the eye, but the opsins are pigments that are associated with nerve cells that allow them to be sensitive to light. Here's the structure of a human eye, viewed cut away from the side. What I want you to notice is that the light would pass from the left side of the slide toward the right, toward that yellow layer called the retina, where the photoreceptor cells are. We have 4 kinds of photoreceptor cells, one each for red, green, and blue, the cones, and then the rods that are sensitive to like grayscale and motion. And they differ from one another because of the varieties of opsins that each of those different kinds of cells has in them. Here's the thing that's crazy. On the left, we have that reference image of the eye, and on the right is a close-up of the retina alone. In this picture, light still goes from the left side of the slide to the right side, and you should notice that the photoreceptors, the rods and 3 types of cones, are on the back side of the retina, not the front. They're on the back side of the retina. So the light has to pass through all of the wiring of the ganglion cells and bipolar cells before it actually gets to the pigmented layer of the retina where the light is absorbed, and that's what turns on the photoreceptors. Your retina is wired entirely backwards compared to the way that you would expect it to be. But notice that vertebrates are not the only animals that have a cameral eye, meaning an eye with a void in the middle of it. Notice that the octopus also has an eye that looks superficially similar to the one that vertebrates have, but in this case, they actually build their retina the right way around, with photoreceptors in the front and then multiple optic nerves coming off the back of it. So why do we have this crappy backward eye? The answer seems to be that if we were going to try to evolve our eyes with natural selection anew, we're stuck with whatever mutations happened before. If we were going to try to unevolve the eye, there would be a lot of steps where the eye would work less well than it does now, and that wouldn't be to our advantage, so selection would tend to work against that. Natural selection is not forward-thinking. It's not goal-oriented towards perfection. Natural selection only does better than, not perfect. Some kinds of vertebrates have developed workarounds to help them increase their visual acuity. The eye on the left side of the slide is from a raptor like an eagle, where they have a structure called the pectin that allows them to superoxygenate their retina so that those cells can recover from their function really quickly, the effect of which is giving them really acute vision. Why are we stuck with this crappy backwards eye? It's just the result of an early mutation that natural selection just kind of dealt with along the way. You know that round plug that's in almost every car that people use for like charging their phone or connecting to whatever? When I was growing up, we used to call it a cigarette lighter. Why is it universal in cars? Why is it that particular direct current voltage? It's because some mechanic somewhere made a decision and it was kept because it worked fine and it's used for a different function now, more for power than for generating heat. But it's kept in the car because it works just fine. The way that natural selection works is that if it's fine, it's left alone. If it's harmful, it'll tend to be selected against, and if it's helpful, it'll tend to be selected for. Natural selection is making these choices constantly. The way that Darwin thought about natural selection was that it was constantly weighing and measuring the sum total of all the attributes of a living thing's phenotype for its survival and reproduction. So how did the eye get built? Early genes for serpentine proteins for nerve cell sensitivity allowed animals to have sensitive spots on their epidermis, and those sensitive spots could thicken up and form cup-like structures in order to develop something that looks like the eye vertebrates have now. What would a simple cup-like eye look like? It would look like this. Those little blue points along the margin of the shell of a clam are its eyes. They're not image-forming eyes, but rather ones that can sense light versus darkness, which is better than no eye at all. They don't need to have that kind of visual acuity when they just need to know when to get out of the way. I mean, here's what they look like when they swim. You don't need a image-forming eye for this. You just need to know where the shadow and where the light is. Why can we see different colors using the kinds of cones that we have? Again, this is due to duplication of opsin genes. I won't get into the details here. You can pause this if you want to read this, but by now you probably get what the plan is. Just to bring things back to the beginning, we started off about introducing genes in organisms that didn't have them before. This is a process that bacteria have been doing almost since the beginning of life. Bacteria can be transformed by picking up loose pieces of DNA in their environment. They can get transduced by the action of retroviruses injecting DNA into the genome, or bacterial mating called conjugation can happen, in which these genes are inserted from a donor to a recipient individual. In each case, the recipient of that DNA now has the information to make proteins that it did not have before. These genes work the same way in a bacterium or a firefly or tobacco plants or in people because cells are dumb and they will do what they are told to do by genes. Some people consider this to be a kind of neo-Lamarckianism. Traits acquired during the lifetime could get passed down to the offspring. Lamarck's mechanism was wrong for his conception of evolution, but in this regard, his basic idea is tenable. But what happens is due to the mechanics of genes and the winnowing effect of natural selection.